Good afternoon. Great privilege to speak to you this afternoon on a slightly different subject at the end of this session, ophthalmology and retina treatment. So I'm Peter Francis. I'm the Chief Scientific and Chief Medical Officer at Ray Therapeutics. We are a San Francisco, Berkeley, and now Irvine, California-based company with a mission to restore vision to patients with retinal degenerations, or put more simply, making blind people see once again with the power of optogenetic therapy. We have lead assets, RTX-015. We've spoken about the data many times, and our 016 program, which is a highly related program for advanced macular degeneration. RTX-015, the initial indication will be retinitis pigmentosa. And RTX-021, which is our treatment for Stargardt's macular dystrophy, which is a common inherited blindness, and also for the broad panoply of patients with age-related macular degeneration of the advanced geographic atrophy phenotype. There are several optogenetic companies working in the vision space. Our key differentiation is that we have the first bioengineered optogenetic medicines that have been truly optimized for human vision. Up until this point, clinical trials have been conducted principally with first-generation or essentially wild-type optogenetic proteins. I'll talk about that in just a second. They haven't really been in any way optimized for human vision, so here we come with the hope to realize the full potential for optogenetic vision restoration, and in particular, taking it beyond restoration of light sensitivity to blind patients now into higher visual functions such as visual acuity, contrast sensitivity, navigation vision, so all of those highly human functions of our vision. And moreover, the first generation optogenetic proteins were never evolved for human vision, and so were relatively insensitive to environmental lighting. So in the lighting that we're looking here at the screen would not be active, and so have had to be combined up until this point with light-enhancing eyewear or goggles, which has a number of challenges, as you can imagine, combining a complex biologic with a complex device, both from a regulatory perspective, but also just in terms of measuring endpoints and patient functionality. We have a very experienced management team. We were formed in 2021, so we've made rapid progress towards clinic. We were delighted to fundraise about a year ago, a $100 million raise. We've also raised money and been recipients of California Institute of Regenerative Medicine grants, CIRM grants, who are very privileged to have progressed our programs with the support of leading venture capital companies as well as CIRM. And I think we would all agree now that the clinical experience, which is more than 10 years with these first-generation optogenetic proteins, has really shown proof of principle, if not proof of concept, that optogenetic therapy works. So we regard our programs now coming in with a bioengineered de-risked protein and an AAV in an AAV vector as being maximally de-risked. And as a result, we feel we have a first-to-market, next-generation approach towards optogenetics. So just a few words about the word I've used several times and how it applies to vision restoration. So really, most of the field is focused on channel rhodopsins. These are naturally occurring transmembrane proteins found in single-cell species. More than 100 years ago, it was noticed that these particular, like algae, had the ability to move to and from light based upon a light stimulus. It was not known until around about 2000 that this was achieved by these unique proteins called channel rhodopsins. So these are standard channel proteins in that they are once the channel is open, they allow flux of anions such as calcium, etc. But they're unique in the fact that the gating of the channel opening is by photons of light. And in fact, they're exquisitely sensitive. One photon of light of the right wavelength will result in channel opening. and maximal opening can be achieved with as little as 2 photons, so exquisitely sensitive. This has really been a neuroscientific discovery field up until this point, because you can encode these channel rhodopsins in a gene therapeutic. You can deliver it to neurons, and then using light you can stimulate those neurons, and you can study pathways, disease pathways, and so on in the brain. What our inventor said about 15 years ago was, if you could do that in the brain, maybe you could do the same thing in the retina, which itself is a neural structure. And maybe the cells, the neural cells that survive retinal degeneration could be made light sensitive. And this is the foundation of visual optogenetics. Thereby, light coming into the eye stimulates these surviving retinal neurons. They send a signal to the brain, which is interpreted as vision. And this has been shown preclinically in multiple animal models by multiple independent laboratories and published many, many hundreds of times to be an effective way of improving at least light sensitivity and restoring light sensitivity to blind animals. And as I mentioned, at least proof of principle now in humans. So where do we fit in terms of optogenetics? We would obviously love to treat everybody with a retinal degeneration, but really we're focused on patients who have advanced disease. So if you direct your attention to the upper schematic here, it's a cross-section of the retina showing the multi-layered structure with the photoreceptors, those naturally light-sensitive cells that we all have. These are lost throughout most retinal degenerations in a progressive fashion, and they do not regenerate. So this is what results in loss of visual field and visual function and ultimate blindness. You can see over time these cells are lost, and so are many of the other interneuronal cell types, all of those remodeling. But these retinal ganglion cells at the bottom of the image survive right up into the end stages of disease. And so while there's some tremendously exciting work going on in terms of gene replacement and CRISPR and so on, you really need those photoreceptors to be present for it to be efficacious. Whereas optogenetics doesn't rely on the photoreceptors, it relies on the fact the retinal ganglion cells are still present, which they are right up to the end of disease. So our initial targets are in that retinal ganglion cell area, and then for earlier patients and potentially for higher resolution vision, actually treating those interneuronal bipolar cells, and I'm going to talk about that in a little bit more detail in a second. So we have a niche, which is a substantial market nonetheless. Maybe 20 to 50% of patients may qualify for optogenetic therapy. So our target indication with our lead product, RTX-015, is in retinitis pigmentosa, targeting those retinal ganglion cells. You can see the schematic of how vision is typically lost. Our estimation, there are a total of 125,000 or so patients in the US with this condition. We think we're in that sort of 20 plus minimum, very conservative estimate of a treatable, addressable market. The rest of the presentation is really going to be focused around Stargardt's and geographic atrophy and our RTX-021 program, which is targeted towards those interneuronal cells with the potential for high-resolution vision, and it'll become clear why that's really important. In these macular degenerations, Stargardt's is a juvenile form, essentially, of the age-related form, which is incredibly common, cause central vision loss with no hope of recovery. And we're looking to actually you see the black spot there in the center. We're actually hoping to restore vision in that area. I'm hoping this might play. This is our lead asset, but I don't have any sound. Utilizing light-sensitive, naturally evolved proteins, channel rhodopsins, to control neuronal function with light. Visual optogenetics can potentially restore vision in people with retinal disease by converting surviving neurons into photo-sensing cells. Most treatments aim to slow vision loss. Optogenetics can restore vision, is not dependent on any gene or mutation, and has the potential to work on all forms of retinal disease, including retinitis pigmentosa, Stargardt disease, and the geographic atrophy form of age-related macular degeneration. Ray Therapeutics has bioengineered the next-generation channel rhodopsin-based gene therapy treatment for human vision, RTX-015. It is administered through a safe, quick, minimally invasive intravitreal injection in the doctor's office and repurposes the retinal neurons that survive in retinal diseases into light-sensitive photoreceptors. The retinal ganglion cells then directly relay visual signals to the brain for interpretation as vision. RTX 015, a major advancement in the field of optogenetics with the potential to dramatically improve the quality of life for those affected by retinal degenerative disease. Hopefully a nice summary of what I just said, and thank you to the audiovisual team for making that possible. With regard to that product, we're excited to announce the inauguration of a central testing facility. So with these patients with poor vision, they really do need special attention. And for too long, they've been going to busy clinical sites where there's not enough time, there's not enough expertise for them to get the best kind of testing. And we recognize in early phase trials in particular, there's a real need to be able to demonstrate and disentangle what is real efficacy in terms of vision at the earliest stage. So we've essentially built a central testing facility. Patients will go to the clinical sites for safety assessments, and then they would come to the central testing facility for this. It will also allow us to develop additional testing capabilities. So our Vision Research and Assessment Institute is based in California, and it's very close to many of our clinical sites, so patients won't have to travel very far. We've staffed it with a team of world-leading low vision experts to really ensure the standardized testing of patients with low vision. So really just a few words now about our second program, which is about a year from clinic, RTX 021, for Stargardt macular dystrophy and geographic atrophy. As I showed you those images before, both these diseases are characterized by central vision loss, which is irrecoverable. As you saw from the video, these treatments can be given with a simple office-based procedure, and in this case, we deliver an AAV gene therapy to target on bipolar cells. These are the on switches for vision, the cell types that stimulate vision. They are topographically arranged in the retina, so there's a point-to-point reference for each one of them. So theoretically, they should have a much higher level of resolution. And we've ensured on bipolar cell expression. As you can imagine, there's also an off bipolar cell system, as well as many other cell types in the retina. We've ensured that by licensing and further developing a on bipolar cell specific promoter, expressing our proprietary optogenetic bioengineered protein crown or Cocha 3M, which is optimized for human vision in the vision range. So this is our hope, is to restore high resolution macular vision to these patients, which will be active in ambient lighting. As I mentioned, it's a macular treatment to on bipolar cells. With our advanced vector technology, we're planning low dose, 1E9 vector genomes total, which is a very low dose in terms of intravitreal, as well as gene therapy. And as a result, we're looking for a safe and durable, we anticipate a safe and durable response. We're looking for visual improvement at an early stage. All of the animal studies, all of the human evidence suggests that you see early efficacy within a few months. And by virtue of higher resolution vision, we're hoping to be in approvable endpoints to speed our clinical progress. As I mentioned, there will be a large market for this, low cost of goods because of small volumes, and so improvement of significant quality of life and activity of daily living to our patients. And how can we justify this? A little bit of our preclinical data in terms of a summary. We work with a specific animal model that is entirely, completely blind and has no visual responses whatever. And then we test that using an animal testing system that mirrors a lot of what we do in human clinical testing, in particular pre-verbal infants, in that the animals are exposed to this rotating bars, and we can change the contrast, we can change the grating width, and in that fashion we can measure things such as visual acuity, contrast sensitivity, and so on in an in vivo system. So I'm not showing you any in vitro evidence of optogenetic function. This is all in otherwise blind animals. So RTX 021 shows exquisite light sensitivity. So just to put this in context, that 1E12, 1E13 photons per centimeter per second is lighting that's lower than this. It's street lighting at night. Many of the earlier treatments were only needed those goggle systems in order to be able to see, and this one will not. It also retains a very broad dynamic range, right up to bright sunlight, so it should be active throughout all environmental lighting. And this is a critical piece of data, really, in terms of this high visual functions. These animals in red cannot see at all. In black, this is the normal mouse vision. We're able to achieve at least 50% creation of vision in previously completely blind animals. For those in the know, these animals also will lack the higher cortical pathways for high-resolution vision. So we think we're actually seeing a bit of a ceiling effect here because of the model. We probably could achieve even higher visual acuity, as is borne out by a complete creation and restoration of contrast sensitivity responses where there were none before. So a substantial improvement in those parameters that would be really, really important to patients with Stargardt's and age-related macular degeneration. So really, we hope to realize the full potential of optogenetic therapy with our bioengineered proteins, and hope for a substantial quality of life improvement for the many millions affected with retinal blindness. Thank you so much.